All right, let's get started with the second half of the session. And first speaker is Sophien Kamoun, who is going to tell us probably about effectors again.
All right. Thank you, Blake. Great pleasure to be here, and really enjoying the day today, and really great talks. So I'll start with some acknowledgements. I see how this works. OK, good. I'll start with some acknowledgements. First, the funding agencies. And I work at the Sainsbury Lab in Norwich. And we're funded mainly by the Gatsby Charitable Foundation, but also acknowledging some people. And in particular, I want to highlight Sylvain Raphael, Liliana Cano, and Joe Wynn, who were the principal people driving the genomics part of the talk that I will tell you about. And then the second part will be about translational research we're doing, and that was driven by postdoc Maria Eugenia Segretin, who's now back in Argentina, also Marina Pais, and then some recent research by Sebastian Schonak, who's also at the conference here. OK, so Ken Shirasu showed you this slide. This is from a paper in Science about a year and a half ago, highlighting big seven pathogens that are threatening food security. What Ken didn't tell you is that 5 out of the 7 are actually filamentous plant pathogens, so fungi or oomycetes. So this is my favorite organism, the potato blight pathogen, Phytophthora infestans. This is an oomycete. It's not a fungus, quite unrelated from fungi, but it has a filamentous type growth, so it does look superficially like a fungus. And then the other 4 include a couple of rust species, rust fungi. And then rice blast is a quite famous pathogen too. These are also a big threat to food security. So a little more about these filamentous plant pathogens. These cause very destructive diseases. Often the destructive diseases are caused by biotrophic pathogens. So that slide I showed you earlier, actually all of those are biotrophic plant pathogens. That means that they require living plant cells to cause disease and colonize the plants. And also, they tend to be host specialized, which means that they only infect a few plant species, one or a few plant species, and not a broad range of hosts. So that's an interesting aspect of these plant pathogens. And the other second point I want to highlight is that the reason they are destructive is not necessarily because they kill plants very effectively. It's really because they're very adaptable. And by adaptable, meaning that they can very rapidly overcome plant resistance in the field, and also sometimes even jump to new hosts. And this is a very important theme and very important in the sort of very important feature behind their success. And then there's also other, I think, aspects of their life cycle that make them very successful. Check this out. This is a slide from Australia. It's a wheat field infected with rust fungus. And this tractor is blowing through it. Look at this cloud of spores. So this is, and we'll tell you more about it in the next talk, but this is also quite a remarkable feature of these organisms. The population sizes are huge. So far we have about 30 genomes described. Of course, there's many sequenced, but now these days people sequence genomes and then they take years to analyze them and describe them. But there's about 30 described, and I have a slide here summarizing these 30 genomes. It's very complicated. Don't expect you to read this. But the point here is we have 3 major groups. We have the oomycetes. So again, this is a very different, very distinct group of eukaryotes, quite distant from the fungi in the tree of the eukaryotes. And then we have fungi here. We have basidiomycetes and also ascomycetes, 2 main groups of fungi. So all of these are plant pathogens. And the part I want to highlight is this column here. This is the genome size, and the circle size reflects the genome size. It's proportional to that. And you can see 3 major groups which have quite large genomes. This is the Phytophthora infestans, the potato blight, quite a large genome size. The second group here are the rust fungi that I just told you about. And then the third group are the powdery mildew fungi here, Bloomeria graminis being an important pathogen on barley. So these 3 groups are all, again, biotrophic, host specialized plant pathogens that happen to have evolved these quite large genome sizes. This is another way to look at the data. This is a plot where we compare genome size to the number of genes. This relationship is quite linear with bacteria. You can see that there's a very nice correlation there. With the eukaryote, we tend to lose the correlation a little bit. And clearly, you can see that the Phytophthora infestans, the potato blight, and its sister species is there. This is Bloomeria graminis, again, the barley powdery mildew. These are genomes that deviate from the norm in the sense that they are quite enriched in non-coding DNA. And this is why you see them deviating from this relationship between genome size and number of genes. So they have fewer genes than they should, given their genome size. OK, so the interesting point here, and really the topic of my talk, is that in filamentous plant pathogens, bigger is better. OK, so this is unusual because, first of all, these are definitely larger genomes than non-parasitic relatives. For instance, in Phytophthora infestans, the genome size is 240 megabase. This is huge. This is twice Arabidopsis. 74% repeats. We recently sequenced the closest non-parasitic relative of Phytophthoras. This is an oomycete that invades dead litter or litter and dead tissue in salt marshes. The genome sizes of those species are about 20 to 25 megabase and less than 1% repeat. So 10% of the genome of Phytophthora infestans. So quite a distinct difference there. Rust fungi also quite large genomes. Powdery mildew also in the hundreds, over 100, 65% repeats. So this is clearly something that has evolved independently in independent lineages, very different lineages, in fact, and clearly a case of convergent evolution in this large genome size. So what's interesting, too, is that this really contrasts with what is really known typically in parasite symbionts. So Saskia told you about phytoplasmas, which have a very reduced genome. But the textbook dogma really is that parasites should evolve to have smaller genomes. So there's many examples here. We have apicomplexans, parasites of animals. They have compact and reduced genomes. There's a plant parasitic nematode here, also another compact genome, smaller than C. elegans. It's very well known that symbiotic parasitic bacteria tend to have extremely reduced genomes, sometimes in a few hundred kbs. And last week in Nature, we heard about the spider mite. It's a herbivore. And this is also the smallest sequenced arthropod genome to date. So the dogma, the textbook dogma, is that parasites should become dependent on the host. They should lose genes and have reduced, simpler genomes. And so this is not the case at all in filamentous plant pathogens. We have bigger genomes, trend towards bigger genomes. So the question we're facing is, what are the evolutionary trade-offs that counterbalance the cost of the larger genomes? And that's a question I will attempt to answer today. OK, so before we get to that, I have to introduce effectors. We heard a bit about effectors earlier. And so effectors are secreted molecules by plant-associated microbes that essentially perturb plant processes. We heard beautiful examples from Saskia there on how sophisticated these effectors can be in perturbing plant physiology. So some interesting concepts here. First of all, a concept I really like is we should really appreciate that effectors are essentially plant proteins. Okay, operationally they are plant proteins. They function inside plant cells. So even though the genes are sitting in the microbial genome, the protein itself functions inside the plant. And that's a very interesting concept, because that means that any natural variation in the sequence of these effectors should directly impact the interaction between the pathogen and the plant, and therefore we can expect that effectors will be directly targeted by natural selection. And then the current paradigm in the field is that studying effectors is important to understanding parasitism. Now they have been reported in bacteria, oomycetes, fungi, and nematodes, and more recently also through Saskia's work in insects. Quickly, some of these concepts. So we have these effectors are now delivered by various pathogens, various microbes inside plant cells. They target plant proteins, so they alter these plant proteins in some ways. And this results in altering plant processes, which essentially should be something that helps the pathogen, helps the microbe colonize the plant. So any effector activity that will help the fitness of the microbe will be selected. But what's also interesting about these effectors is that they can also chip on the wire, if you like, and they can activate plant immunity. So this happens when they're recognized by plant immune receptors. These are the same immune receptors that Blake told you about this morning, the NBLRR proteins. When the NBLRR proteins detect the effectors, this can happen directly or indirectly, then there is effector-triggered immunity. There's a plant cell death response that is activated and that basically overcomes the activities of all the other effectors, resulting in effective resistance in the plant. So you can imagine now how these effectors are under conflicting selective pressures. If they land on the good host, on the susceptible host, then you expect the effectors to evolve to be more efficient on their targets and helping the pathogen colonize the plant. But when they land on the wrong plant, a plant that carries a receptor that recognizes one of the effectors, then the effectors becomes now liability for the microbe, and there is now selective pressure to get rid of that effector to overcome resistance on the plant side. So obviously this creates a very dynamic situation in terms of coevolution between the microbe and the host. Okay, so where do these effectors reside? And I'll show you some slides from our work with Phytophthora infestans. We have a very large effector repertoire. There's about 1,000 of these effector genes in the genome of the pathogen. Phytophthora infestans has about 18,000 genes, so it's quite a lot of the genome is devoted to effectors. And what we discovered is that the effectors tend to reside in these expanded repeat-rich regions. So this is the alignment or collinear regions between Phytophthora infestans and two other Phytophthora species. This genome is much larger than the other two, and the black bars are the repeats. You can see how there's a bunch of effector genes here shown with red arrows that are in the middle of these repeat regions and these expanded regions. We still have regions that are quite dense in genes, where the genes are tightly packed with little repetitive DNA, but the genome is populated with these repeat-rich regions. Sylvain Raphael in the lab developed a method to visualize this by looking at intergenic distance. So for every gene, you can measure the 5' and 3' distance to the neighboring genes. And if we do that with our Phytophthora infestans genome, we find a very unusual profile. This is very different from what you would see with most organisms. This is Plasmodium, rice, human genome. That usually there is a uniform, more or less uniform distribution, certainly not a bimodal distribution like we see, where many genes are within 1.5 kb from each other, but we have a whole bunch of genes that are in the middle of the repeats. And what Sylvain did here is he highlighted some of these effectors. These are the ones that are recognized by immune receptors, by plant immune receptors, and you can see that they all sit in these repeat-rich regions. And the trend is true for essentially over 95% of all the effector genes. So since I'm talking about other organisms, there's similar situations with fungi. So this is the genome of Leptosphaera maculans. This is a genome that 65% repeats. And in this case, the repeat-rich regions are quite gene-poor. There's only about 150 genes in Leptosphaera that are in the repeat region. So you don't see the density there. But you can see that the known effectors are inside these repeat-rich regions. There's 3 of them that are highlighted in this particular graph. So this essentially highlights a general trend in filamentous plant pathogens where effector genes populate plastic regions of the genome. So we have the Phytophthora example I just showed you. So this is like an example of one effector gene in 100 kb of expanded repeat-rich region. This is the Leptosphaera. In Leptosphaera, they actually have isochore-like regions. So this is a GC plot. You can see these drops in GC. So these are 80-rich expansions that are present throughout the genome of Leptosphaera maculans. That's where the effectors locate. But we also have other examples of plasticity. This is rice blast. One effector of rice blast of Magnapothia oryzae is called AVR peta. And this is present near telomeric repeats at the chromosome ends. You can see how the gene can be embedded within the telomeres or can be within essentially tens of bases from the telomeric repeats. Another example of plasticity in these filamentous pathogen genomes is Fusarium and other fungal pathogens that carry conditionally dispensable chromosomes or accessory chromosomes like these 2 here, which tend to be horizontally transferred between species or strains. And that's also where the effector are. This is work from Martijn Rep in Amsterdam. You can see there's a bunch of effector genes. They're all on this conditionally dispensable chromosome, which also determines host specificity when it's transferred from one strain to another. So again, a very interesting theme that is recurrent in these organisms where these effector genes populate plastic regions. Okay, so quickly some experimental work we did. So this is really highlighting also that these pathogens, the host-specific ones, we don't find cases of coevolution. This is one case of how these pathogens co-evolve with the host. They track the host, so as the host evolve over time, speciates, the pathogen will also track the host. We don't see that. What we see is, in most cases, is host jumps. So we see the pathogen infecting host and occasionally jumping to a new host, and we end up with a very dynamic situation where the pathogen is continuously moving from one host to another. This is certainly the case in Phytophthora infestans. This is the clade we studied. Basically, these are the species that are close to the potato blight. P. infestans here and its sister species infect completely different botanical families. The ITS region is essentially identical between these, so these are recently diverged. Clearly case of host jump type of evolution, and they also specialize on their respective hosts, so we don't see cross-infection when we test these strains on different hosts. So a very obvious model is that we expect that the host jump to have a very dramatic impact on the effector genes. And the reason is that these effectors function inside the plant cell, right? Remember that. So they have a target. And so we can expect that in many cases, when the host jump occurs, these effectors now are sitting in a plant cell that's from a different botanical family. So we expect the targets to either be missing or be quite divergent. So we expect either the effector to become useless or to adapt to that new target. And indeed, when we sequenced the genomes of this clade of closely related species, we discovered that there's clearly an excess of pseudogenized and genes with evidence of positive selection among the effectors, much more so than in the remainder of the genome. So really to summarize this work, what we think we have now is a two-speed genome, essentially, what we like to describe a two-speed genome. Gene-sparse regions of the genome, which show high rates of structure and sequence variation, and with signatures of adaptive selection. This is an example of one of the plots we generated. So if we take these intergenic distance plots and then in the bin then put the number of genes that show evidence of positive selection, we clearly see that the genes in the repeat-rich regions are enriched in genes under adaptive or positive selection with higher non-synonymous to synonymous substitutions. So our model is that the gene-sparse regions, they underpin rapid effector evolution. They probably underpin host adaptation. And our favorite model is that this probably explains how these lineages of pathogen can persist over long evolutionary periods, which is true because these are very deep lineages of plant pathogens. So our favorite model at the moment is really taking ideas from the clade selection concepts. And this is a fantastic book by G.C. Williams on that topic. And what we think is that the strains, the lineages that have the ability to jump hosts more frequently or adapt to new hosts more frequently are going to persist over macroevolutionary periods. And they're less likely to go extinct because if you are not jumping and adapting rapidly, then at some point your host population either will go extinct or will become fully resistant, and you will have basically a lower likelihood of persisting over long periods of time, and you will go extinct. So this is an interesting model that I would love to discuss with some of you about how best to test it. One idea is to look at lineage age and compare that to genome size and see how that actually compares and challenge this model in that way. Okay, so in the last few minutes, I'll tell you about some of the translational work we are doing. And this is basically looking at how we can explore our knowledge about the effector biology of this pathogen to engineer durable resistance. I just told you that these pathogens are very good at adapting. So do we have any hope actually to engineer resistance that will last in the field? But the classical approaches have been blind to the pathogen. Basically, breeders have introduced these resistance genes without any knowledge of the effectors, without any knowledge of how these genes are functioning in terms of the molecules that are perceiving. So one idea here is, do we have an effector set that is more conserved than the remainder of the effectors? And this could be potential Achilles heel that we can exploit in developing resistance. So we've done a lot of sequencing. We sequenced the major strains that are causing epidemics. We have about 6 or 7 genome sequences right now of the pathogen, including both genome sequence and transcriptional profiling. And one example is this strain called Blue 13. This is an asexual lineage that exploded in the UK. This genotype now accounts for 70 to 80% of all the isolates. It's more aggressive. You can see how it spread rapidly. That's an example of a strain we sequenced. We also sequenced a strain that caused an epidemic on tomato in the US in the last 2-3 years. So the outcome of this is that even though we have 550 effectors in this particular class, so the genome has about 550 genes, we only found about 50 that are maybe a core set that is always conserved and always expressed in at least these 4 strains that we looked at. So we do think that we have a core set of effector genes such as these. This is to show you that they are indeed expressed and that there are immune receptors that recognize some of these genes. So perhaps these resistance genes will cause more durable resistance. I don't know the answer to that, but certainly I know that if we eliminate these guys, if we don't consider the genes that are recognizing the dispensable effectors, if you like, we will maximize our ability to maximize the potential for durability in the field. So I think the approach here is more filtering out the R genes that clearly don't work and don't last. Okay, so I'll tell you briefly about another way to exploit this knowledge, and this is about one of the core effectors called AVR3A. AVR3A is recognized by an R gene and BLRR gene called R3A. What's interesting about AVR3A is not very polymorphic. There's 2 major alleles in the P. infestans population. It's always present, always expressed, the core effector. And when we knock down AVR3A, we also have reduced virulence, so we think it's important for the pathogen. But R3A recognizes only one of the 2 alleles. So it recognizes this allele here, Ki, but doesn't recognize EM. So what Maria did is she did a PCR-prone mutagenesis, error-prone PCR mutagenesis of R3A, generated library of about 2,200 mutants of R3A, and then screened for recognition of the EM version of AVR3A. And indeed, she recovered a mutant like this one here, which not only recognizes Ki, like the original R3A, but it also now recognizes the other allele of AVR3A, the EM allele, which the original R3A doesn't. So she now identified single-site, single amino acid changes in R3A that expand the recognition repertoire to all the known alleles of the effector in the pathogen population. So this is obviously a very useful gene that recognizes all strains of Phytophthora infestans. And then the surprise was that we also discovered that this gene, although it comes from potato originally, some of the mutants in R3A can recognize members of the AVR3A family that are occurring in other Phytophthora species. So this is an example from Phytophthora capsicum, which is a pepper pathogen. The original R3A does not recognize this homologue of AVR3A, but some of the mutants we have do recognize this particular homologue. And more recently, Sebastian Schornack got interested in Phytophthora palmivora, and maybe some of you saw his talk, his poster about that. This is a pathogen that's destroying oil palm plantations in South America, particularly in Colombia. 200,000 hectares were lost in a couple of years. It's very dramatic, and with all kinds of consequences on oil palm production, biofuel, and also the drug trade. And what we did here is this very wonderful, actually, sequence, and really a very nice example of translation genomics. We sequenced the genome of the Colombian strain of Phytophthora palmivora, identified a large family of AVR3A homologues, screened the R3A plus family, and then discovered that we have some of the AVR3As can be recognized by some of the R3A mutants. And this slide is not showing up very well, but what you see here is when we make transgenic plants with the R gene then we have resistance against at least one of the Phytophthora palmivora strains. And now we're looking at which combination of our genes can give us the widest spectrum of recognition to engineer full resistance against this nasty pathogen. So, okay, so I'm calling this next generation resistance breeding because the idea here is that we are exploiting actually knowledge about the pathogen biology and effector biology to engineer new resistance specificities. And what I like, of course, that this is really stemming from basic research, from basic knowledge on the effector. And also one potential consequence of this is that we could consider non-GM solutions. And you'll hear more about Effectors tomorrow in Ulla Bonner's talk. But if you haven't heard about them, then please go and read the papers, because this is quite an amazing technology with huge potential for establishing genome editing in plants. In particular, there's this paper here that showed with zinc fingers that zinc finger nucleases can be delivered through virus vectors transiently in plants to enrich for mutagenesis particular loci. So in a non-transgenic way, because the gene is never integrated in the genome. So I'm happy to discuss with some of you about this, but I think this has potential applications for manipulating our genes and generating synthetic R genes. So time ran out, but this is my last slide. I think the vision here is that we have, can we develop framework to rapidly generate new resistance specificities and introduce this into crop genomes? Essentially, can we deploy new resistance faster than the pathogen can evolve and turn the arms race basically in an arms race between the biotechnologist and the pathogen? So my belief is that silver bullet solutions will not work against this type of pathogens, and we need to essentially continuously generate and have a pipeline to continuously generate new resistance specificities. Okay, so I'll stop there, and I want to thank my colleague at Sainsbury Lab and then also collaborators, various collaborators we've had over the years. Thank you very much.
Okay, we have time for some questions. Yes, Magnus at the back. Is there a microphone?
I'll just be loud. Okay. So, so we're talking, when you say never bet against the pathogen, right? So this might actually be a bar level question, but that was a recent paper about pollen utilization and fungal invasion, right? So essentially, suggests that basically the fungus is a strategy there, right? I mean, you can easily become resistant, right? But then you're sterile, so what's the point, right? But if one could help the crops this way, maybe they can actually come up with a strategy that the fungus couldn't defeat, if you know what I mean.
I mean, that's obviously an interesting question, right? If there are some host susceptibility factors that the pathogen absolutely requires,
right?
And can we perturb these? I think that could come from this work on the effectors, and actually not now on the immune receptors, but on the effector targets and manipulating those effector targets. The problem is that so far, a lot of these, including the paper you're referring to, these tend to be quite important proteins in the plant and that you can't easily manipulate. And so you would think that if it was actually something quite dispensable on the plant side, there would have been mutants that would have emerged that made the plant resistant. So again, I like this kind of concept of clade selection, because maybe at the end, the pathogen that made it into today's biota are the ones that actually went after really important critical components on the plant, and the ones that actually exploited maybe surface receptors or other components that are not so important in plants would have been purged out and never made it into the modern biota. So
sure.
That's a way to look at it. But yes, that's another angle, and that hasn't been looked at that much.
Yeah, you show the effectors in the ocean of repeats. I wonder just what is the nature of the repeats? Is it just the AT rich, low complexity, or some
all sort of stuff?
AT-rich?
So it varies a little bit between different species, but usually it's a mixture of what looks like functional transposable elements, and then a lot of degenerate and nested TEs and all kind of degenerated repeats. In Phytophthora, it seems like it's ancient, because the classes of transposable elements are quite similar across the Phytophthora. In Leptosphaera maculans, for example, where there's the isochore-like regions with the AT-rich regions, it looks like there was a recent invasion of that genome by a limited family of retrotransposons, which are AT-rich. So that explains why these regions are AT-rich. So that looks like a very amazing actually situation where a fungus get invaded by transposons and then turn into a nasty plant killer, right?
So what is the mechanism of actually mutating the effectors? Is it a REAP program or just a recombination of the...
That's great, because actually this talk is inspired from a review we just submitted last week, which I encourage you to read. It will be probably Nature Reviews Microbiology soon. And we cover this topic. It's a fantastic topic of are there actually mechanisms to increase mutagenesis. And one very nice example also in Leptosphaeria is where in ascomycete fungi you have the ripping. So some of you heard of ripping, this repeat-induced mutagenesis. And this is essentially a mutagenesis process that is triggered by repeats and obviously happens a lot in transposons. And many of the transposons of Leptosphaeria have been degenerated by ripping. The problem is that ripping occasionally will actually slip through and leak to neighboring sequences. And what they discovered in Leptosphaeria is that there's an effector, an AVR effector, that was pseudogenized, and that was pseudogenized by ripping. So in this case, the fact that it was located next to transposon increases likelihood of being pseudogenized. And that's really a fantastic example of another reason why they might benefit by being close to transposons. We really don't understand this. Another possibility is obviously RNAi. They could be now exploiting an epigenetic process that is targeted against the transposon, but now that the effectors are actually exploiting to be turned on and off. And my colleague Mark Geisen has beautiful studies showing that in Phytophthora, soja in this case, soybean pathogen, virulence can evolve through epigenetic silencing of an avirulence effector. In addition to that, you have, of course, the structural instability and the increased recombination that these regions create.
And was there one last question, Ulla? Did you? No. Magnus, another bar question? Or,
no, this is actually a great question. What about, are there cassettes? Are there a horizontal gene transfer? I mean, you might imagine. Right,
yeah, okay, so in Phytophthora infestans, there's not good examples of recent, at least recent, relatively recent AGT. But I told you about these conditional chromosomes. Yeah. That's a very clear case of horizontal gene transfer. And there's also a case of a disease, actually, that was arose in wheat. It's a fungal disease, Stagnospora, and it arose in wheat through horizontal gene transfer of a single gene, a toxin gene, that was transferred probably in the 1940s. And because of that, the new pathogen that acquired this toxin became a new pathogen, new disease on wheat in the US. And so there are cases also of AGT.
Sophien, I wasn't convinced by your argument about the core set of effectors. Why would the plants not have discovered that the pathogens have a core set? So the corollary to this would be, so it wouldn't make sense to me if these would evolve as fast or even faster than the non-core effectors.
Yeah, I went too fast, so I didn't define what we mean by core effectors. So we don't mean non-polymorphic ones. There is no non-polymorphic effector. So we have now all these genomes, right? There's not a single one that we believe is a really strong candidate that has no amino acid replacements. So our definition is the gene has to be intact, and it has to be expressed, induced in planta like they all are. So that doesn't rule out polymorphism. So for example, we treat AVR3 as a core effector, although it actually has evaded recognition by the plant. So just clarification.
All right. Thank you very much, Sophien.